Amy, I know that this is a very important topic for you. How are you and Vincerx addressing the historic ADC challenges around bystander effect and toxicity?
Yeah, bystander effect. That's really, that's really one of the main things that we've been thinking about when it comes to challenges, the historic challenges of ADCs. And like I mentioned in the beginning, developing the right kind of linker for the right kind of situation. There are times when bystander effect can be part of the mechanism of action. Maybe with these lower expressed targets, and you do want systemic release of your payload. But as a company, we've really focused on trying to eliminate that non-specific release by the linker, because we believe that's associated with more systemic toxicity. So we've developed a linker that is only cleaved by an enzyme called legumine. And so that amino acid sequence that we've built into the linker can only be cleaved by legumine. It's not going to get cleaved by a pH enzyme or just a generic protease or a cathepsin. It can only get cleaved by legumine. And so we believe that's one way to address that. I think Sandra will probably talk about her antibodies and making sure her antibodies are getting to the right place. I mean, it's definitely, there are multiple ways to think about it, but we've really purpose built our bioconjugation platform to overcome the bystander effect with a specific linker and cleavage process.
Yeah. Linker technology is amazing to me. I was reading an article just recently about a photosensitive linker technology where they introduced like UV light to create the a cleavable linker that's only susceptible to UV light. I've read other new upcoming linker technologies that, you introduce I guess it'd be a fourth element, right? Like a chemical element to release the payload at a specific time. Joe, I mean, what are some of the more exciting linker technologies you're seeing? I mean, I just mentioned a couple of them, but.
Yeah, I think there's some appreciation as well, or some work happening, not to the same extent as cleavable linker inside the tumor, but outside the tumor and tumor microenvironment. There's a lot of innovation there. I mean, there's also an appreciation of the immune system, playing that role in supporting ADC responses and clinical responses. So I think there's a lot of TAMLin cleavable linkers outside the tumor. There's a lot of pH sensitive, not in the endosome, those are potentially are even not even internalizing ADCs. They work on the surface. So there's a lot of linkers coming up online. I think something that could potentially be interesting is companies working on multi-payload approaches, because that could potentially combine multiple technologies and innovation into one. You can talk about different linkers going on a single ADC, right? Half of your payloads could be outside the cell doing whatever they want to do, and then half of your linkers would cleave inside the cell and then be toxic, right? So you have that like I'm not saying a company is doing that, but there's a lot of things early on or stealth mode that are still being cooked to address the innovation and to innovate on the linker side that could potentially have different sites and different mechanism of actions. I mean, the other thing, I'll be short, but the other thing is the payload. I mean, we're touching also, and I think that a lot of companies are working with payloads that we don't necessarily know their full MOA yet, even though that is still in discovery and preclinical, especially discovery. We don't know the MOA, but then they're seeing that diversification in the payload that potentially could be an issue with the aggregation, could be an issue with the linker, could be an issue with non-specificity. Those are early days. But then if I want to link it back to the manufacturing, that is also going to put a strain on CDMOs or even companies, right, like Vincerx and Mythic, where we don't even have tests and QC tests to test those ADCs in the manufacturing process. So that actually sophisticates testing and manufacturing as well. So there's the payload and linker, outside the cell, inside the cell, testing not existent today at least, or being developed. So that is also an opportunity for that.